All right. Hello, everyone. Thank you so much for being here this morning for the Georgia Tech Neuro Seminar Series. My name is Yasmin Basile. I'm a neuroscience PhD candidate in Dr. Michael Borich's lab over at Emory, and I'll be introducing our speaker today. It's my honor to introduce Dr. Til Ole Bergmann, an associate professor of neurostimulation at the Johannes Gutenberg University Medical Center in Mainz, Germany. At the university, Dr. Bergmann is the deputy head of the Neuroimaging Center and principal investigator of the neurostimulation group at the Leibniz Institute for Resilience Research, which focuses on studying the neurobiological correlates of emotional memory processing for resilience. Dr. Bergmann is a biological psychologist and cognitive neuroscientist by training, interested in the function of neuronal oscillations and cognition, in particular for the organization of information processing and the gating of synaptic plasticity in the awake and sleeping human brain. His methodological focus is on the simultaneous combination of non-invasive transcranial brain stimulation techniques with neuroimaging and electrophysiology and the development of novel approaches such as state-dependent brain stimulation. Dr. Bergmann is also the recipient of the prestigious John Rothwell Award by the BrainBox Initiative to acknowledge and reward excellence in non-invasive brain stimulation research. Today, Dr. Bergmann has traveled from Mainz, Germany to present his talk titled Combining Transcranial Brain Stimulation with Neuroimaging for State-Dependent Stimulation and Causal Network Interrogation. A quick note for questions, if we could save all questions for the end of the talk, there will be a brief 15-minute Q&A session during which we'll walk around and pass around the microphones if you have a question. All right. With that, please join me in welcoming Dr. Til Ole Bergmann.
Thank you very much. Great. Thanks, Jasmine. Thanks, Michael, for having me here. Thanks, all of you, for coming to this meeting and listening to the work I'm going to present. So yeah, I'm a psychologist by training, but I'm like working in systems neuroscience for about 17 years or so, mainly with brain stimulation and in combination with imaging, EEG, fMRI, and everything I can combine. And I'm going to talk about these different transcranials and non-invasive brain stimulation techniques. I'm going to give you a bit of an overview in the beginning for those of you not familiar with the techniques. And then I'm mainly going to talk about TMS, so transcranial magnetic stimulation, and its combination with fMRI and EEG. And if I have time left in the end, I'll also talk a little bit about transcranial ultrasonic stimulation. That's basically the new kid on the block when it comes to non-invasive brain stimulation techniques. If I'm talking too fast or you can't hear me, just let me know. Okay, the presenter stopped actually working. I'll just restart. Should I just... that was it. Okay, thanks. Good. So transcranial magnetic stimulation in a nutshell. So what we do is we have a copper coil basically in a plastic housing. It's a double coil that's held over the scalp. And for the fraction of a millisecond, the strong current is running through that coil, producing a magnetic field reaching into the brain, where it induces again an electric field in the brain tissue, which is then exciting axons, causing a full depolarization and action potentials. So it's eventually an electric stimulation technique, and the magnetic field is just a Trojan horse to bring it painlessly through the skull into the brain. If we stimulate the motor cortex, then transsynaptically, we're going to excite these large pyramidal cells in layer 5, which project down to the spinal cord and then onto the muscle. Where we can measure with surface EMG electrodes, the so-called motor evoked potential, the MEP. That's the working horse of the TMS community, because it's the amplitude nicely shows it's proportional to the number of cortical output neurons that you excite above firing threshold. So there are at least 3 synapses. So it's already a network stimulation effect. Outside of the motor cortex, we can quantify the brain response by, for example, transcranial, so by TMS evoked EEG potentials, TEPs, which can be derived by multi-channel EEG recordings at the same time. I'm not going to talk about those a lot today. just to show you like how TMS works in principle. Then with another technique, I'm not going to talk about at all, just for the sake of completeness, transcranial electric stimulation, TES. Here we use weak currents injected via 2 or more electrodes attached to the scalp. We're talking about a few milliamp and a few volt only, and it distributes widely across the cortex. If you use direct current stimulation, here are some early recordings in animals from the 60s that show the principle nicely. Depending on the polarity, you can increase the spontaneous firing, or you can decrease the spontaneous firing, but it's not triggering action potential. It's not like TMS that you evoke an action potential. It's more changing the threshold for spontaneous activity to increase or decrease. If you do this with an alternating current, the idea is that you can entrain oscillations, and it works nicely in vitro and also in animals, because there you can reach high enough stimulation intensities where it can entrain firing, for example. In humans, it's a bit more difficult because we're somewhat limited with the intensities. So we're like at borderline efficacy. Sometimes you get very nice effects. Sometimes it's just not strong enough to produce any entrainment effect. And then the third technique, which just emerged in the last few years, well, it's also much older. The first attempts have been in the 30s, but it's now re-emerging as a technique for neuromodulation in humans, is transcranial ultrasonic stimulation with ultrasound transducer, which focuses the kinetic energy of ultrasound waves in humans between 250 and 650 kilohertz. And it focuses it superficially or also deep in the brain. And depending on the type of transducer and the frequency, you can have different sizes of these slightly cigar-shaped foci. For example, with 500 kilohertz, about 3 millimeter width, 2 millimeter length. So this is much more focused than what we can do with TMS, let alone TES. Here, for example, an attempt to focus it on the thalamus in humans. And well, depending on how you position your transducers around the cortex, it can be really millimeter precision, or we're talking about this cigar-shaped, slightly elongated foci. A lot of the energy is absorbed by the skull, it's scattered by the skull, it's refracted. So it's the skull is our enemy here. TMS can overcome the skull easily, but focused ultrasound really has a problem going through it. But it's possible at these lower frequencies, and one just has to do proper simulations. I'll come back to that later, hopefully if I have time. And the idea is that, yeah, stop working again. That most likely it works via the activation of mechanosensitive ion channels. Might also work directly mechanically on the membrane, and there are other mechanisms discussed, but most likely it's working activating mechanosensitive ion channels, and there's calcium influx in the cascade that eventually also causes action potentials. So it's also a potentially super threshold technique, but it's not an immediate synchronization. It takes a few hundred milliseconds possibly. So these are the three main techniques now for non-invasive transcranial stimulation in humans. And you can do a lot of different things with it. So in principle, with online or offline approaches. Online means that you stimulate and you look into the immediate response of the brain to the stimulation and its behavioral consequences possibly. And it comes in different flavors, so you can use it to quantify certain properties of a network, excitability, connectivity, etc. Stimulating strong enough to get an output in MEP or TEP or bold response. And I'm mainly talking about that today. Or you can interfere with ongoing or task-related brain activity and disturb a brain region relevant for certain cognitive performance. And then you see a change in reaction time and in error rates, etc. Or you can just more gently modulate the timing or the level of activity by rhythmic stimulation, for example, with TMS or TACS. And then there are the offline effects, where a prolonged protocol, repetitive stimulation, induces ATP or LTD-like synaptic plasticity, and thereby after effects that outlast the stimulation by minutes to hours. And again, you can image those later on, EEG with fMRI, with behavior, etc. And we try to improve the targeting of these methods, the selection of parameters in terms of where, when, and how to stimulate, also based on imaging information, running function localizers with fMRI, EEG, MEG, doing using the individual anatomy and E-field modeling to know exactly where we want to stimulate. Use, for example, evoke potentials, or also the online tracking of ongoing EEG activity to target specific events. I'm going to talk about that today, to increase the temporal precision. And in principle, we could also individualize our stimulation by taking information from the participant about, let's say, the frequency of specific oscillations, to then adjust our stimulation protocols accordingly. Non-invasive brain stimulation is used a lot in cognitive neuroscience to answer questions about which brain regions or processes are causal for specific functions. And quite often you will read in papers simplified statements like, okay, we use certain brain stimulation technique to change neural activity in region X, and that changes cognition or behavior. So this region is causal for that certain function. But of course, that is oversimplified, and behind that is the implicit assumption that your brain stimulation technique produces an E field, if we talk about TMS or TS, in the brain, in the target region, which then changes local neural activity, which then maybe also cause remote network effects, because it's always a network stimulation, right? Which then changes information processing, neural processing in the brain, so the basis for cognitive function, which eventually can be measured by a task with certain behavioral measurements. So if this chain of causation with all these cause-effect pairs, and only if all these red arrows here are true and realized actually in the correct way, then you can make that claim. At the same time, you have some off-target activation. You're stimulating regions that you didn't mean to stimulate, co-activate them because it's not as focused as you would like to have it. And then you're stimulating networks you didn't mean to stimulate, effects on other cognitive functions, or the same you're measuring, right? And you create confounds. Or you have peripheral co-stimulation confounds. The TMS makes a click when the coil discharges, and you feel it on the scalp. You might activate some scalp muscles. TS causes a tingling sensation on the scalp, might even cause phosphines with TACS, etc. All these things activate your brain, and thereby have also effect on brain functioning and on your causal inference. And then you have different task demands, brain states, which I'm going to talk about, etc., which have an effect on all of those. So it's tricky if you want to derive your actual conclusion, right, about causality here, especially given that all of these variables, and most of them are latent, they're not directly observable, right? The E field we can simulate. It's very difficult to measure in humans, in healthy living humans. For the neural activity, at least we have neuroimaging, right? And this is also why I'm going to talk about that particularly today. The cognitive process, again, we can model, and we can have good psychophysics to measure the behavioral response. For focus ultrasound, we have the same problems, just smaller. I mean, we have a smaller focus, but we can also co-stimulate regions that we would not like to stimulate. And also there are some auditory confounds I can come back to at the very end if I have time. So the imaging is important because it allows us to create proof of neural target engagement, that the brain region we meant to stimulate actually got activated, and to see the network that also got activated. And maybe there are some brain regions that explain our behavioral effects even better than the node of the network that we started stimulating. So we have all these different imaging techniques, EEG, MEG, fMRI, FNIRS, PET, etc., which is slightly different spatial and temporal resolution here. So the spatial and temporal resolution, but they're all at a similar level. And we can combine different techniques like EEG and fMRI to get the best of both worlds, the good temporal resolution of EEG, the good spatial resolution of fMRI. And luckily, we can also combine them with the causal brain manipulation techniques, the NIPS techniques. And many of them we can combine online. So this is technically demanding. It's creating a lot of artifacts in the imaging modality, and I could talk just days about these artifacts. So if you have questions, feel free. I'm just going to skip over the technical difficulties in this talk, because otherwise I would get stuck pretty early. And I'm going to start with TMS fMRI. So that's our setup in Mainz. We use MR receive coils and a 7-channel surface coils, 2 of them, so 14 channels. We can flexibly position them and have our TMS coil here actually on top. So we're stimulating through this ultra-thin coil, which gives us good signal to noise ratio at the cortex level, slightly worse deep in the brain, but there's always a poison you have to pick. We have online stereotactic, frameless stereotactic new navigation using this head trackers that are even visible when the subject is in the center of the bore, so we can continue tracking the TMS coil relative to the head while the subject is in the MRI and is being scanned. The question is like, why should we do that? Main reason is we're able to show proof of target engagement, both at the cortical level, where we stimulate, but also at the network level in deep brain regions that we might stimulate indirectly. Maybe that's even our main purpose of the study. We can also map effective connectivity. Stimulating one region, getting a response in another, shows you that there's an actual connection. It's not functional connectivity, it's effective connectivity, right? It's directed also. We can change stimulation parameters like the intensity or the coil orientation and see which works best empirically for our subject in principle. We can investigate state dependency. It has been shown many times that the network activation in particular depends a lot on what the subject is doing at the time of stimulation. So the activation is rooted into different networks depending whether you're processing a stimulus at the time or not, or in which state you are. And it helps us also to interpret our data, especially because we can see the confounds we are creating. I mean, you see the auditory cortex activated with TMS click. In the EEG, you also get auditory confounds, but it's more difficult to tell from the technique. We can see activation due to expectation of upcoming stimuli of sensation, etc. And when we find some behavioral effects of our stimulation, we might find a brain region in which the activation or the TMS-induced activation correlates much better with the behavioral effect than in our original target. So we can actually pinpoint the where the stimulation effect in the brain happens that's behaviorally relevant. I'll give 2 examples here from our ongoing work. One focusing on the motor cortex, for which it has been shown already, like in the early 2000s. Well, there's a nice activation under the TMS coil where you stimulate, but only at super threshold intensities. So it could be just you need enough energy to get a bolt response, or at high intensities at the motor cortex, you get a muscle twitch, right? And then you get reafferent feedback, which activates your sensory motor cortex. And there has been work that tried to somehow indirectly figure that out, but we thought, okay, let's solve that by blocking that route. So we used an ischemic nerve block, taking a blood pressure cuff, inflating it to the systolic blood pressure, and keeping it like this for about 50 minutes, 50. So after about 25 minutes, the block is complete, and TMS is not able to produce any MEPs anymore. They disappear in red. SEPs, like measured with EEG, disappear. So there's no information going in any direction. And then we have these 15-20 minutes time to make measurements. So in this study, so we do this thing in the MR as if it wouldn't have been already uncomfortable enough. First, just to show that our MR coils, which are a bit special, are sensitive. Well, if you do finger tapping with nice activation, entire sensory-motor cortex in the entire motor network, so that works. Then in one session, we want to show that the ischemic nerve block actually works. So we do electric stimulation of the median nerve at the wrist at an intensity that gives you also muscle twitches and activation of the sensory-motor cortex. And then we block it and redo it. And what you see is, well, first we get sensory-motor activation due to these electric stimulations. During the ischemic nerve block, it's gone, and that's the difference. Okay, so the block works. Now we do this with TMS. We stimulate the motor cortex. By the way, here is a beautiful auditory cortex activation, right? I mean, from the TMS click, it's even louder in the MR because of the magnetic fields interacting. And then though we stimulate at 120% of the vestibular threshold, so clearly above threshold, so you would get MEPs and twitches if it wouldn't be blocked. So it's almost completely gone. There's a little bit of activation in the postcentral gyrus. But most of the difference is most of the motor cortex activation is removed, which means actually all the studies reporting suprathreshold effects in the motor cortex under the TMS call basically show reafferent feedback. So it's good to check and be careful when you work at the motor cortex because of, well, one of our nicest output feature, the MEP, master twitch, is also big confound. Now moving into deep brain regions. So imagine we would be interested in stimulating the subgenual anterior cingulate cortex, for example, as a target for the treatment of depression, or some other regions. So we are interested in the VMPFC for fear extinction, memory consolidation. Then we can assume that there's a polysynaptic link from some cortical sites. And there's been a lot of work from the community interested in treatments for depression. And Michael Fox and Robert Cashin, these guys have worked out that if you look at resting state functional connectivity with fMRI, and you take the JSSC as a target region, you get these correlated and anti-correlated networks. And you can personalize the treatment to basically by selecting the target coordinate as the coordinate with the voxel with a maximally anti-correlated function connectivity with this deep brain region in the subgenual ACC. The problem is, well, and it actually, the closer you are to this optimal point, the better the treatment response. And this has now been integrated in some accelerated protocols like the SAINT treatment protocol that has been developed in Stanford. So people are using this kind of individualization now. The thing is, it's a single coordinate, and the coil orientation is a bit unclear. And we simulated the E field induced by the TMS in standard orientation. And we actually did it for a lot of different points and coil orientations, and each time evaluated the field. And you see that with the standard orientation, we produce a field that's not so nicely overlapping with your target. Your target should be the anti-correlated cluster. In fact, you're stimulating a lot of the positively correlated one. So we then basically optimized the coil orientation and position for the induced electric field, which is the effective part of the stimulation, to maximally overlap with the, let's say, negatively correlated ones, while trying to maximally spare the positively correlated ones. And this results in very different coil orientations. So the approach is basically we calculate for our deep brain target, it's the VMPFC, our function connectivity map, individualized, get the negative function connectivity target, also get the positive one, but because we actually wanted in the study to see whether they do have opposite or different effects on the deep brain regions, because that's not empirically shown yet, it's an assumption. To our E-field optimization, position the TMS coil on the participant, and then actually also select control targets, one for each cluster, one for the positive, one for the negative, which are maximally uncorrelated. So it's a very nice control condition, because we just move the coil a little bit to a region where it's, the E-field is mainly stimulating voxels that have no connection, no functional connection with our target. But we create a similar noise and sensation. This is also why it needs two different ones, because depending on what you here on the side, you might get more jaw muscles, it's more unpleasant compared to the more medial sides. So we have two matched control sides, uncorrelated as nicely matched as possible, right? It's never perfect. And then we determine the stimulation intensity, also based from on the E-field modeling. And then we actually stimulate the subjects at that selected orientation and position. We do get a positive board response under the TMS call, and this time there's no reafferent feedback. It's the DLPFC, not the motor cortex. And in this case here, there's a single subject, pilot subject, in the VMPFC, or a bit more anterior, we get a negative board response. And this is actually already the contrast. So the negative FC target with the respective control target. So this removes all the auditory confounds, all the smarter sensory confounds, all the unpleasantness, maybe the anxiety related to being in the MR. So all those effects are matched, and it's only by moving the coil a little bit to a different connectivity target. So we are currently running the study. I think we're with 14 or 16 subjects, but I don't have the data yet. So I hope that soon I'll be able to show that. What I can show you already is that the matching worked for the first 16 subjects. So we have in the loudness ratings for the two different clusters, negative and positive, and then the actual target and the control. And they're not significantly different. Also, the unpleasantness rating is similar. And even the stimulation intensity, even though it could result in different stimulation intensities, because we would choose for the control target an intensity that causes similar unpleasantness, similar loudness, but it might not result in the same physical intensity, but that's secondary. Okay, so hoping that the main sample also works out, we have a method by which we can indirectly target deep brain structures. Now, actually, okay, just checking the time, switching gears and going to EEG, which has the advantage of being like fast, has a high temporal resolution. And I'm mainly going to present work where we did not use it as an output, but as an input to decide like when to stimulate. Because the brain has been treated for a long time as a static black box. So we just stimulate at some point, randomized, and we get an output, let's say MEP or whatever, which has a lot of variability. If we would treat the brain as the dynamic system that it actually is, with a lot of fluctuating brain states, and we would sort our trials according to the brain state at the time of stimulation, we might get very different outcomes. And I'm interested in neural oscillations, and I think they also define brain states, global brain states, but also local cortical states. They're often generated in corticothalamic loop, and they're also, depending on the oscillation, different structures involved. They're under control of neuromodulators, particularly noradrenaline, dopamine, and so forth, which are relevant for changing actually the brain state with a dominating frequency. You close your eyes, or when you get relaxed, you have alpha oscillations, you get drowsy, it disappears again, theta kicks in, and then light sleep, sleep spindles, deep sleep, it's largely synchronized cortical activity, slow oscillations, well, typically not coma, but that would be even more synchronized. And interestingly, but not in the scope of this talk, with the increasing synchronization, you also have a loss of consciousness happening. Neural oscillations are involved in many different cognitive and motor functions, almost any one function, and they're dysfunctional in many neurological and psychiatric disease. So why is that the case? Why are they important for everything? Because I believe they are the major building blocks for computation in the brain. And just to briefly go over some common principles, so with this rhythmic input-output gain modulation, so no matter how an oscillation is generated, you have a more excitable and a less excitable state typically. And in the more excitable state, it's depolarized and excited. If a stimulus comes in, then those neurons will most likely also transmit the information via an action potential. While in the down state, when it's hyperpolarized or actively inhibited, maybe incoming action potentials will not result in action potentials. So it's a discretization of information processing in time. And this has consequences. So if you have neuron populations that are aligned in phase, they can talk to each other because their duty cycles overlap. If they're out of phase, they cannot communicate effectively because the one population is not listening when the other one is talking. So that's the idea of communication to coherence. You have hierarchical cross-frequency, phase amplitude coupling, which meaning that in the more excitable state of a slow oscillation, the amplitude of a faster oscillation is increased, and in the more excitable state of that, the amplitude of an even faster oscillation. So, for example, delta theta gamma coupling, or slow oscillation spindle ripple coupling. So it's a nesting of faster and faster oscillations, which always increase in amplitude in the more excitable state of the slower oscillation. Or more sophisticated ideas like gating by pulse inhibition, which proposed for alpha oscillations. So with increasing amplitude, there should be increasing inhibition happening, which is like rhythmically suppressing bottom-up gamma oscillations, which are transporting like sensory information upstream. And there's phase coding, phase precession in the hippocampus. You know, this from this red neural navigation experiments, or we've postulated also alpha gamma phase precession when scanning brutal scenes. And something I will come back to, phase amplitude coupling for phase-dependent plasticity. So if you have an, let's say, a slow oscillation during sleep in the depolarized upstate, and then a sleep spindle arrives exactly at the most excitable, most depolarized state, so that the thalamocortical input into the apical dendrites of these cortical neurons, when they're maximally excitable, then you have calcium influx and can produce ATP like plasticity. If they would arrive during a different time, different phase, where they are not excitable, you might not get ATP or even ATD, long-term depression. Okay, so we used a technique called like real-time EEG triggered TMS to investigate some of these questions. Here it's about pulse inhibition, which has been shown or mainly hypothesized for the visual system. We investigated this for the mu alpha of the sensory motor cortex. And the question was, is it actually pulsed facilitation, pulse inhibition, but with increasing amplitude, with increasing inhibition, but there's always a phase where there's zero inhibition, which is the same as if there would be no oscillation present. Or is it pulsed facilitation, where with increasing amplitude, you have asymmetric increase in pulsed facilitation, but there's always a phase angle where the facilitation is as low as if there would be no oscillation, or is it even symmetric? Boring. So what we did is we targeted the lower 20% of the individual mu alpha, basically the oscillation is desynchronized, not present, not observable, or the upper 20% with a full-blown nice oscillation. So we analyzed it in real time and then stimulated, and random phases if there's no oscillation. But if there's an oscillation, then the peak, falling flank, trough, or rising flank randomized. And we measured excitability with motor evoked potentials in motor cortex. And we used a paradigm called short interval intracortical inhibition, SICI, which uses a second conditioning pulse at low intensity, which is inhibiting the MEP to the second pulse, and has been shown that it's GABA A receptor mediated. So we wanted to see whether there's any GABAergic effect happening if it's pulse inhibition. So the targeting worked out well. We do see the mu alpha peak only for the high power trials. Here's a type frequency representation. It's local at the left sensory cortex, where we want to target based on our EEG montage. So it's not some occipital alpha bleeding in, it's a local one. And most importantly, well, on the average, we don't see any oscillation in the random condition, but we do see targeting of the peak, falling flank, trough, rising flank, when we targeted those specific phase angles. So this processing has to happen fast, we're able to target them. And what we found is that there's actually not a single phase angle where we do see an inhibition relative to our randomized low amplitude condition, but there are the trough and rising flank where we have a facilitation. So each time, like once a cycle, there's a facilitation from baseline. So we can investigate like basic new physiological questions with this technique. There's no change in SICI. So it looks like pulse facilitation in the motor cortex, well, we measure with MEPs, but be aware that our EEG montage is very sensitive for actually the post-central, the somatosensory mu alpha where it's generated. So it's maybe that in that somatosensory cortex, we do have actual pulse inhibition, which results in pulse disinhibition once per cycle of the motor cortex. And we are running studies at the moment to figure that out. My colleagues from Tübingen have we showed this basic difference between peaks and trough of mu alpha first. They have exploited it by repetitively targeting either the less excitable peak or the more excitable trough in one session with triplets of 100 Hertz TMS, again and again in the peak or again and again in the trough. And if you hit the more excitable trough again and again, then afterwards you have an increase, lasting increase in the excitability. But that's not the case if you have the same amount of stimuli, same intensity, but targeting a different state of the oscillation. So this is phase-dependent plasticity, which could potentially be used also for different oscillations in different parts of the cortex, and which make sure that we actually do have this phase-dependent plasticity mechanism in the brain, and we can mimic it with these phase-dependent plasticity protocols. This is the first study of this kind, which I did as a PhD student. Actually, when Michael was visiting in 2009, we figured out in Kiel, where I did this with Hartwig Siebner. We were basically recording this data, published in 2012, so it's a while ago. We targeted the slow oscillation. It's slow, so it's an easy target, right? So with the methods were available, but then it was possible. In red, the slow oscillation up states, in blue, the down states. Half of them we stimulated, half of them we just set a marker and did not stimulate. So we did this during sleep in this very comfortable semi-upright position, had fixated, neo-navigated, noise masking on the ears. And these are the TMS evoked potentials. Well, in red upstate targeted, in blue downstate targeted, and you see that the average evoked potential is well, the slow oscillation that we targeted plus something that rides on top of it. So we had to subtract the unstimulated trials, and then we get here with the different scaling, the TMS evoked potential, right? Which looks very different than the same stimulation during wakefulness. It's basically, again, a slow oscillation. You stimulate the brain during sleep. It reacts with the oscillation that it can produce in that state anyway. We also measured the motor evoked potentials again. And while during sleep compared to wakefulness, there's a general reduction in excitability, which could be at the spinal level even. But it was also systematically different between the up and down states. So during the hyperpolarizing phase, it was smaller. So and during the depolarizing phase, it was larger for all subjects. And the TMS evoked potentials also showed an effect, like at the stimulation site locally was larger the EEG response when we stimulated during the up state. And also when spreading like slow oscillations do over the entire cortex, it was still larger when was an up state at the time of stimulation. And this is not an all or nothing phenomenon, but we could actually do single trial correlations. So looking at the slow oscillation amplitude at the time of stimulation, which predicted the MEP amplitude, but only from channels at the stimulation site. For the TPs, we can't do single trial analysis, the signal to noise ratio matter, but we can bin them, and then we see the same thing that the so when there's a larger ongoing oscillation, then the TP is also larger. Now, 10, 12 years later, we moved from slow oscillations to spindles. So Umar Hassan, now former PhD student in my lab, he managed to detect spindles in real time. Now, this time it's not in a reclining chair, but in the bed. Again, neo-navigated, targeting spindle-free periods during sleep, different phase angles of the spindle, and then after the spindle. And again, targeting worked out as planned. We're stimulating spindles and not some artifacts or adjacent frequency bands. And due to our montage, it's relatively local. And what we see here is similar pattern, but different, which means it's again like trough and rising that are like comparably largest, but actually not different from the baseline, from the spindle-free time periods. And it's again the falling flank that is the lowest, but this time this is the outlier. So the it's a pulsed inhibition happening once per cycle to one specific angle of the spindle, which means we can now in the future with projects planned where we want to repetitively stimulate during the spindle at the time when it's most excitable and produce plasticity, for example, in the motor cortex during spindle. So opening a window of plasticity in the sleeping brain which could be a very interesting approach also for neuro rehabilitation. It's a totally unused 8-hour time window, where the brain is doing a lot of plasticity, a lot of maintenance, a lot of consolidation. So if one could tap into those mechanisms and utilize it, that would be very interesting. Of course, we also plan to study memory reactivation and more at the level of motor learning or also declarative memory. So I've shown you a couple of examples of how we can use for different oscillations this approach using TMS with EMGs, the MEPs as a measurement, and using the EEG to inform us about the particular brain state, in this case, amplitude and phase of an ongoing oscillation. But you can also think about more complex protocols in the future, more complex brain states. So we've already made the transition from open loop brain state independent protocols to open loop brain state dependent protocols. It's still not completely closed because we track alpha or slow oscillation spindles, and then we measure the MEP, but we do not change the spindle necessarily. At least it doesn't look like that. A closed loop would be when we change the variable we're monitoring. Then we have a closed loop that we can change a brain state from an undesired to a desired, from dysfunctional to a functional brain state, and maintain it. And we're not there yet. We have done some software developments to help this kind of research, to facilitate it. The Brain Electrophysiological Recording and Stimulation Toolbox, the best toolbox, it's an open source MATLAB toolbox, which allows for a lot of standard protocols, finding the motor hotspot, automatic closed loop threshold detection, input-output curves, everything, but also all the brain state dependent EEG triggered protocols, also some TMS, fMRI, synchronization, and so forth. So it helps to solve some of the technical problems. But so we can measure in real time and do the calculations and trigger the stimulator. But the thing is, we need to identify those brain states in real time, also more complex ones, connectivity, entropy, some sophisticated network analysis, whatever you think is interesting as a brain state. In the case of EEG trigger TMS, remove the artifacts in real time. That's the tricky part. If we want to stimulate maybe each cycle of an oscillation, that's not possible at the moment. And the most difficult of all, like, how do we change a brain state? Which part of the network do we need to stimulate when and how to shift from one attractor state to the other, from an dysfunctional to a functional one? How do we keep it there? So maybe control theory is going to help us, but this is, I think, the biggest problem. If all the technical thing is solved, like, how do we change the brain? How do we make it behave like we would like? So this is actually the biggest problem. So actually, I hit the 45 minutes and I haven't talked about ultrasound. If you're fine, I would add another 5 minutes at least to give you a rough idea. I'm not going into all the details. I have more slides. If you're interested, I'm happy to talk about that afterwards. But just to give you a quick idea, you can do a lot of different things with transcranial ultrasound. It's used in neurosurgery already for ablation with high intensities. You can heat up the tissue to 45, 60 degree. You can ablate it. If you do this for the VIM or STN, you can treat basically tremor with the subject walking in and out afterwards without opening the skull. But it's ablation. Or use the low intensities with simultaneously injected microbubbles. You can open the blood-brain barrier. That's typically a good thing. It keeps stuff out of your brain that shouldn't be in there, but it also prevents important pharmacological treatments to enter the brain because molecules are too large. They can't pass from the bloodstream into the brain tissue. And if you have these microbubbles and you excite them with ultrasound, they get into stable cavitation, they oscillate in size and press against the tight junctions of the epithelial cells, and then it opens them transiently, and simultaneously injected molecules, pharmacological compounds, or what have you, can actually get from the bloodstream into the brain. With a grant now to actually explore this in mice and also in the future in humans. And then neuromodulation, which is more what we do. And I'm not going into all these other fancy things. That's the typical HIFU setup with high intensities, high intensity focused ultrasound. This is what you would do for ablation. We keep the temperature below 1 degree in the brain, so it's not a thermal effect. Most likely a mechanical effect or the ion channels or the effect in the membrane. We apply the treatment in a pulsed fashion. So not going into details here, but for not too much energy to be deposited at the same time, not too much warming to happen, we only give it a certain amount of time, like, let's say like 30% of the time it's pulsed at, for example, 1000 hertz. And then different kinds of protocols can be created, which actually opens up a huge parameter space. And at the moment, the community tries to figure out which parameters do what, how do we get excitatory or inhibitory protocols. So we are not there yet. Depending on the type of transducer, we can have smaller or larger foci. We can control them if we have multiple elements. We can steer them in distance, for example. That's the simple system that we are using, which is a 4 elements annular array, with which we can steer between superficial and deep brain regions, but just in a straight line. If we couple it with some ultrasound gel, which is tricky, because it needs like direct coupling to get from the transducer to the bone. That's the tricky part, into the brain. Navigation is key, right? Because if you tilt it a little bit, then in 8 centimeter depth, you might, like with a 3 millimeter wide beam, you might just be off. So we try some robot-based new navigation, but we also try and use it in the MR directly. Or to the relative light, which to try and build individualized 3D printed adapters and attach it to the head. And then you have to do acoustic simulations based on bone imaging to see where the beam is actually ending up and plan your position accordingly. Bone imaging is key. CT would be optimal, but in healthy participants, we can't do that, right? Because of the radiation. So we have to use MR, which is not optimal for bone imaging, but we try to improve that. And then, of course, as always, we would combine it with other techniques for proof of target engagement, with EEG to see whether we can change oscillations, with fMRI to see whether we get a bolt response or modulation of task-related bolt, and with TMS in the motor cortex, just because you always start at the motor cortex, right? To use MEPs to measure excitability of the motor cortex while sonicating and modulating the excitability. Yeah, just some ongoing work in the MR. This is our TMS focused ultrasound setup. We stimulate through the transducer. We need a large coil because it's so far away from the head now because of the transducer. And there have been 2 publications showing that there's a suppression of motor evoked potentials if you start sonicating a few hundred milliseconds before the TMS pulse. Long story short, like 2 years later, 3 labs working on that. But a lot of different experiments. This is now on bioarchives and soon to be resubmitted. The suppression was an artifact, unfortunately. So ultrasound is inaudible per se, by definition. But if you pulse it at 1000 hertz, then you hear a 1000 hertz tone, and it's conducted by the bone, and it's in a weird way. You might have shear waves in the bone, etc. And then you hear a sound that's located somewhere in your head, and it's really difficult to mask. We tried masking with bone conduction headphones and everything, but so not going through this in detail. The thing is, across different labs, we tried control sites, stimulating the white matter instead, stimulating only with noise, no ultrasound, and well, we always get this suppression of the MEP. So it was in that case not a transcranial effect. The animal work is pretty clear and promising, and also there's some good work in humans now, but we still have to figure out the right parameters and control these peripheral co-stimulation confounds by ramping pulses, by good masking noise, to make sure that we're actually stimulating, or that we're actually measuring only true brain stimulation effects. Okay, I'll end here. Sorry for going a bit over time, but I wanted to show you the exciting ultrasound stuff we started to get into. very different from the electric and magnetic stimulation. I hope that I've convinced you that non-invasive brain stimulation is a very powerful tool and a diverse tool to not only like neuromodulate, but to also just perturb and measure to characterize brain networks in combination with fMRI, in combination with EEG. We can read out particular characteristics of these networks and study brain function in health and disease. And yeah, particularly this simultaneous combination, which is a bit of extra effort, but it allows you to look into the actual neural effects of the stimulation and not just hope that you would affect the brain at the position where you put your TMS coil. So I thank all my collaborators and my team and funding agencies, of course, and thank all of you for bearing with me. I'm here for a few days, so there's a lot of time for questions, actually. Thanks a lot. Oh yeah, and I should mention I have a postdoc and PhD positions to fill. So if you're interested or know someone who might be, let me know. Should also be on the website. Yeah.
Thank you. Wow, that's a great talk. I have a lot of questions, but I'll start with just one. You showed with the pulse inhibition with alpha. Sorry, not pulse inhibition. You asked whether it was pulse inhibition or pulse facilitation, and then it's facilitation, at least for the motor evoked potentials. Do you think that generalizes to sensory processing and alpha?
I don't think so. So I think the reason why we do see facilitation or maybe disinhibition is really that we measure from the motor cortex, but that our oscillation might be extracted from the sensory cortex. So with one study, not actually completed, but not yet analyzed that part, where we also did just some other sensory evoked potentials, EEG potentials, median nerve stimulation, time up to the peaks and trough of the mu alpha, to see whether maybe the incoming efferent input into the sensory cortex, whether that might be modulated in the opposite way. So whether there's actually pulse inhibition that during the trough, there might be maximal inhibition happening, and this maximal inhibition causes then a reduction of the sensory to motor inhibition. So it's a disinhibition, a rhythmic disinhibition of the motor cortex. But this is speculation, so we don't have the data yet. There is quite some existing data on actual detection performance, sensory detection performance, and its relation to mainly alpha, mu alpha power, but it's very mixed. So there's positive, there's negative linear relationships, and there are inverted U shapes. But the phase has not been investigated yet. So that's the first time we're doing that. Yeah.
Thank you for the talk. I have just one question. You mentioned that you didn't use a noise masking during the TMS experiments that you present right now. Am I wrong?
So we did use noise masking.
For both?
Yeah, we used them for all these experiments. The thing is, they're most important for TEP measurements if you want to measure the EEG response, because there's quite some overlap with auditory evoked potentials, and which also produce earlier but also late components N100 P2, which you also see for the TEP. So you would need to have successful noise masking to be really sure that what you're measuring is not an auditory confound. The problem is you hardly ever get this fully masked. Also, there's bone conduction. It depends on where you stimulate. Some central sites you can actually stimulate with some coils with proper masking that you can't hear it, but quite often it is audible. And then it's a bit tricky to interpret. The earlier components are easier to interpret than the later ones. since we use most of these experiments here, MEPs as a measurement, they are not affected, right?
Actually, there is the work by Capozio et al. 2021. It's human brain mapping, probably the journal, that shows that MEP is affected by the click, by the TMS click. Is a recent work,
But not the single pulse MEPs, but the click of. So if you have pair pulse protocols, I could imagine that, especially for the long interval cortical inhibition for the LICI protocol, where you have a first pulse 100 milliseconds before. There, I do believe that there is an auditory effect. But I mean, you can measure the volleys running down the spinal cord if you've implanted electrodes like milliseconds after the TMS pulse. At that time, there's no way any auditory information can have reached the motor cortex to have an effect. So I think for single pulse MEPs, we're fine. For pair pulse protocols, I agree, there may be issues.
okay, thanks.
I figured I'll have many. It is loud, Annabelle. Yeah, I feel like I have many times to ask you, but I feel like with everyone here, I'll just ask like a big picture, two big picture questions. One, your experiments are very elegant and rigorous, and to the point of it's almost challenging to extract the sort of the where you take the information from there. And you did make some mention of next steps, but could you talk a little bit about where you see these sort of approaches when it comes to maybe designing interventions, for example? And you kind of alluded to it, but maybe for those who are kind of trying to figure out what you could do with this type of elegant brain stimulation imaging, and as it relates to maybe clinical conditions or something along those lines.
Yeah, I mean, so I think in basic science, we have to first try and figure out like how we can optimize these approaches maximally, right? Be as specific as possible, rule out all the confounds, and then test whether like which steps of personalization of state-dependent stimulation, increasing specificity, which of them are like important, which are not. So how much an effect size will actually change if you do that or not, right? And especially when it comes to clinical use, then of course, it really needs to pay off, right? For example, in this work where we are now trying to optimize these individual targeting for deep brain, like indirect deep brain stimulation. So we have a study plan. So well, grant is resubmitted, crossing fingers that we get the funding to actually in patients with depression, use the accelerated treatment protocol, the same protocol, at least a few days of that, do TMS fMRI before and after with our specific like call orientation, optimization, etc. And then do also the treatment with and without this extra E field modulation. Because a lot of colleagues ask me, like, for good reasons, okay, do we actually need to do this? It decreases the likelihood that we can actually use it in the clinic. Mm-hmm. And so it's a fair point. So we'll have to check whether it's worth it. If it has a significant effect, like in terms of like effect size, if more people that respond to the stimulation because you're on target in each individual and not just 30% of them, right, then it might be worth it. Especially because all you actually need is a structural MRI and a 10-minute resting state scan. And the rest is processing that can be streamlined, that can be put into the cloud, that could be done quickly eventually, right? Mm-hmm. But first need to figure out whether it has a benefit or not. So I always like to cite the study from Alex Sack, who looked into, okay, what is the benefit of using more precise targeting for like interference with the mental rotation task, which they had well established in the lab, and they used either so it depended on the right posterior cortex. So they would either for an individual functional fMRI localizer to find exactly the right spot in each individual, or went for anatomical coordinates or just MNI coordinates or electrode position P4. That's what you would do if you have no clue, if no MRI, just measure and assume C3 is the left motor cortex, P4 is the right parietal. And then they acquire data and say, okay, how many subjects do we need to get a significant effect? And it was like 5 for the fMRI localizer. It was like 40, 45 if you just went for electrode position P4. And then you can do the calculations. Is it worth acquiring so many more subjects to get your effect? If you have the chance to pay for an MRI, and if it's about clinical treatment, of course, then the question would even be, is it ethical to do such a bad targeting? I mean, I understand that you want to make the treatment like available for many people, but if it then only works in a small percentage, because most of the time you're like off target with your simulation, then I would say it's not really ethical to do that, right? So it's a trade-off, and it depends on the case. So long answer.
There's a question on Zoom from Cyrus. Cyrus, if you want to unmute yourself, then you can feel free to ask.
Yeah, can you hear me?
Yes.
Yep.
Yeah, so I was thinking, is it possible to, after you take the CT and have an image of the brain, that you can kind of modulate the ultrasound so it targets a big area, such as the default mode network, and then that it perturbs that specific region?
Yeah, that's a very good point, because so you're asking actually for a less focused ultrasound, right? And that's exactly a point I've been discussing with some colleagues recently, because the big benefit of focused ultrasound is it's so focal, right? We can stimulate the amygdala, or a small, in principle, if we get the targeting right in the bone imaging, we can target small subnuclei eventually anywhere. But if we want to stimulate the cortical region, is this small focus going to make an effect or not? It may even be that our output measures, like an EEG, we might not see an effect because the neuron population we synchronize is so small. So in principle, yeah, we would, one could.
But the default mode network is a large area, right?
Yeah, it's huge. And there's multiple sites, right? Medial prefrontal,
right,
which is splenial. And so you could simulate multiple regions. You could use like 4 transducers to simulate different sites, but each of them is large. So each site would need a larger simulation. I mean, so far there is no multi-site, multi-transducer work. I know of colleagues playing that at the moment. So it's definitely going to happen. But like, if you want to make the beam larger, the focus larger, the problem is that the energy is dissipated, it's distributed over a large area. So you would need more energy that passes to the bone, and then you heat up the bone. So
what about if you cover a large area with several small focused regions? You lay out like a carpet of
yes,
like perhaps 20 different stimulations all over a cortex.
Yeah, that's a good idea, and that's also what, for example, Insight Tech thought. This Israeli company, he had 1,024 transducers over like a hemisphere. You have to couple them all to the skin, which means like in those subjects here, the hair has to be shaved, which is fine if you go for one treatment to get your essential tumor basically ablated, and you go home and you regrow your hair. If you want to go like repeated times for neuromodulatory sessions for treating depression or whatever, you might not want to shave your hair. So, and then the coupling gets really difficult. So the disadvantage of large transducer arrays is the coupling, but I'm sure that the field will work this out. So it's a very young field, and the good thing is, like, based on the physics, we're far away from the ceiling, right? And so it's a lot of engineering questions to be solved. So it's actually a good place to be here to pitch some of those problems.
Thank you so much. Very interesting talk.
And I think that was all of our time for questions, but one more round of applause for Dr. Berg.
Thank you.
Thank you so much.